Welcome to this explainer. Today we're diving deep into the absolute foundational science behind DNA inheritance. We're going to explore an epic, microscopic battle that's happening right now, literally inside your cells. It's this massive biological tug-of-war that dictates exactly how traits are passed down from one generation to the next.
You know, it's a question we've all probably thought about at family gatherings, right? Why do you have your dad's eyes, but you definitely didn't inherit his hair? We get specific traits, sure, but we aren't just exact carbon copy clones of our parents. Well, the answer to that mystery actually lies in the delicate balance of two completely opposing forces working inside our DNA.
Here's how we're going to break this down. First up, the rule of stability, or linkage. Then, we'll look at the engine of chaos, recombination. After that, we'll check out the historical proof with Morgan's fruit flies. And finally, we'll wrap up by calculating variation.
All right, let's jump right into part one, the rule of stability, better known as linkage.
So sometimes nature really just prefers to keep things exactly as they are without mixing them up. And that brings us to linkage, which is basically the tendency of genes located on the same chromosome to be inherited together. Think of it like a deck of playing cards, right? But imagine some of those cards are literally glued together. When it's time to deal, those glued ones absolutely have to be dealt as a pair. They travel together. Well, in the science of DNA inheritance, genes sitting on the same chromosome are exactly like those glued cards.
Because they're physically tethered to one another, genes on the same chromosome do not show independent assortment. For instance, if you have a gene for a plant's seed shape, let's say it makes a round seed, and a gene for seed color, like yellow, and they are sitting on the exact same chromosome, they are fundamentally linked. They want to travel as a package deal straight into the next generation.
So, how strict is this rule? Well, that's where we see the difference between complete and incomplete linkage. With complete linkage, there is absolutely no crossing over whatsoever. The genes are completely locked, meaning the offspring will only show parental combinations. But, nature likes a little variety, and that brings us to incomplete linkage. This is where the rules get bent a bit. Crossing over occasionally occurs, so you get those original parental combinations, but you also get entirely new, mixed-up recombinant combinations.
Which perfectly sets up part two, the engine of chaos, or recombination.
This right here is the opposing force in our biological tug-of-war. Recombination is the formation of new combinations of alleles, and it happens due to crossing over between linked genes during meiosis. Going back to our card analogy for a second, recombination is the mechanism that properly shuffles the deck. It forcefully breaks that glue holding the cards together, which allows entirely new biological hands to be dealt. Honestly, without this, there would be vastly less variation in the whole world.
Let's break down the microscopic mechanics of how this actually goes down step by step. First, homologous chromosomes. Those are the matching pairs from each parent. They align in a process called synapsis. Second, during a really specific phase known as the Pachytene stage, these chromosomes literally exchange segments. This is the famous crossing over. And finally, as a direct result of this swap, brand new recombined chromatids are formed.
Visualizing this process is actually pretty incredible. Picture two distinct chromosomes, one red and one blue. During synapsis, they pair up. Then comes that crucial crossing over step. A physical exchange happens right between the non-sister chromatids. The red chromosome grabs a piece of the blue, and the blue takes a piece of the red. So by the end, we don't just have solid red or solid blue anymore, we have mixed, recombinant chromatids. It's just a stunning example of how our biology actively rewrites itself. The aftermath of this crossover event is really where the magic happens. Instead of just getting two basic parental types of gametes, we actually end up with four distinct types. You still get the original parental combinations, capital A, capital B, and lowercase a, lowercase b. But because of that crossover event, we also get two completely new recombinant types, capital A with lowercase b and lowercase a with capital B. This is it. This is the engine that successfully generates our genetic variation.
All right, let's move on to part three, Morgan's fruit flies, and how scientists actually proved this theory.
So now that we've got a handle on the microscopic cellular mechanics, let's talk about how scientists actually proved this epic tug-of-war was happening. Enter T.H. Morgan. He conducted this legendary experiment using Drosophila, which are just fruit flies. He crossed two very specific types of flies. One had a gray body and long wings, and the other had a black body and vestigial or shortened wings. Basically, he wanted to see if these specific traits were linked or if they'd just shuffle completely independently.
Well, the results were totally groundbreaking. Morgan observed way more parental offspring and far fewer recombinants. The traits didn't just mix evenly down the middle like you might expect. And this proved a vital concept. Genes sitting on the same chromosome definitely show linkage. They really want to stay together, which explains why there were more parental types. But, and this is a big but, because he still saw some recombinants, it proved the linkage wasn't absolute. Recombination was occasionally sneaking in and breaking the link.
Which brings us to our final section, Part 4, Calculating Variation and the Math Behind the Genetics.
So, how do scientists actually measure the outcome of this constant battle between linkage and recombination? Well, they use a remarkably straightforward formula. Recombination frequency simply equals the number of recombinants divided by the total offspring and then multiplied by 100 to get your percentage. By just looking at the offspring of an organism, scientists can literally count exactly how many show new, shuffled traits compared to the whole population. It's a pretty beautiful way of turning biological chaos into clear, predictable mathematical data.
But what's really fascinating is what that resulting percentage actually tells us about the physical DNA itself. There's a direct spatial relationship going on right on the chromosome. If you calculate a higher recombination frequency, it actually means there's a greater distance between the genes. Think about it. If two genes are sitting really far apart on a long chromosome, there's just a lot more physical room for a crossover to accidentally slice right between them. On the flip side, a lower recombination frequency means those genes are physically sitting closer together, which makes it way harder to break them apart.
But here is arguably the most mind-blowing part of the math, 50%. That number is a critical, hard limit in the science of genetics. Literally, no matter how incredibly far apart two genes are in a chromosome, their recombination frequency will absolutely never exceed 50%. Even if genes are on entirely different chromosomes, they assort independently, and they still max out at that 50% chance of recombining. It is this immovable boundary that's just built right into the very fabric of our biology.
So let's quickly recap the core takeaways here.
First, linked genes are structurally located on the same chromosome, and they naturally tend to be inherited together as a unit. Second, that stability gets disrupted by crossing over, specifically at the Pachytene stage of meiosis, which is what causes recombination. And third, this entire mechanism of recombination is absolutely vital. It produces the brand new gene combinations that are responsible for the vast diversity of life. It's the exact reason why you might have your dad's eyes, but a totally unique combination of other traits.
It really makes you wonder, doesn't it? If the absolute engine of our biological variation is strictly capped at a 50% limit, what other hidden mathematical limits are quietly dictating our genetic potential? The rules of inheritance are this incredible, delicate dance between order and chaos, and we are honestly just scratching the surface of understanding the choreography. Thanks so much for joining me on this explainer, and keep questioning the science behind what makes you, you.